Thursday, at the start of lecture, your second assignment from Dr. Bolter will be due, and in order to help you get prepared, in order to answer questions, we still have some office hours. All of them are going to be down in the lab in A456 Franz. And Tuesday, tomorrow, 1 to 2, David Chow is going to go. David Sherman will go 3 to 4. Wednesday, 1 to 3, I'll be on, and David Sherman will clean up 3 to 4 on Wednesday. So plenty of time still to get in there and ask questions. All right, we're going to start a new module now, and so I'm going to start this by telling you a true story. So I've got a whole pile of little girls at home, and they try to engage me in their play. And usually they assign me to the role of the evil fairy queen. And I hate being the evil fairy queen because I'm really bad at it. And they say things like, oh, Daddy, the evil fairy queen would never say that, or the evil fairy queen would never do that. So the other day I was really delighted when they got out rubber dinosaurs. I'm like, oh, cool. I know how to play rubber dinosaurs. This is one of my favorites when I was a little boy, you know. And so they like hand me a Triceratops, like, yeah, all right, I know exactly what to do this time, cool. So I got my Triceratops scouting around for danger, and one of my other daughters has got a T-Rex. It's advancing on my Triceratops. I'm like, oh, this is cool, I know exactly what to do. I've got my Triceratops pawing the dirt in anticipation of this interaction, and then her T-Rex fell upon my Triceratops, and with its stubby little arms started hugging my Triceratops and then started kissing my Triceratops. I tell you this story to point out the fact that there are big sex differences in behavior, and I was ready to play the little boy version of rubber dinosaurs, and obviously there's a little girl version of rubber dinosaurs that I knew nothing about. Now, in the case of human beings, our behavior, you can find lots of sex differences in behavior, but we're always left with a question of what is it? Is it culture and rearing, or is it biology that underlies these sex differences? And I think it's important that we not fall into categorical thinking, as is now kind of popular, that it's all culture and rearing, as I was taught when I was about your age, or that it's all biology, as you may be hearing now. The pendulum has sort of swung in the other direction. But indeed, for humans, it's an interaction. But for science has actually gone a long ways in being reductionistic and looking at the elements that could underlie that interaction. So if we're going to look at the biology of sex differences in behavior, obviously we can't use humans, both for ethical reasons and for reasons that we have culture. And, you know, the culture could either diminish or exacerbate biology, okay, or create sex differences where there is no biological basis. You can't really tell. But if we're interested in sex differences in behavior, we've got to turn to models. And the model that I have used for many years now is the zebrafinch, here shown slightly larger than life size. And here's the male here on your right. You can see it's got kind of ruddy orange cheek patches, zebra barring for which it's named, and you can see a little bit down its side. It has these brown and white spotted feathers. And the female lacks this plumage. She doesn't have the cheek patch or the barring or those feathers. So they're sexually differentiated on the outside, and also they have a dramatic sex difference in behavior. Like most birds in temperate climates, the male sings and the female does not. Now, the female isn't mute. She will make sounds, but she will never, ever engage in this behavior that we call song. And I'm going to give you a few examples here of song behavior. Come on, guys, get in here and let's do it. Okay. So I'm going to play some songs that were recorded from 3 different males, and I'm going to, I don't know if we can hear this, but we'll give it a shot. Okay, be real quiet. You hear that even in the back? Okay. I'm going to play another male now. Okay, one more time. First one. Second. And the third. Okay, so you can see their songs have some similarities, but also each one is individually different. And that's because they learn their songs. And here you can see in the shadow of nest box, this is a little, whoops, this is a little fledgling. And about this time when the little males pop out, they will pay attention to their father, and they won't make an exact copy of their father's song, but they will make a song that is similar to his song. And how do we know this? By rearing studies. This is one that I actually didn't mean to do, but ended up doing. I had a bunch of these birds at home, and this pair mated and had babies, and about the time they fledged, which would be about the time the little male would be learning the song, the dad died. Okay, so he only had his mom as a model. So even though you've only heard 3 zebrafinch songs, I'm going to play. This is Leslie. We can't tell what sex they are when they come out, so we gave them kind of genderless names. But this was Leslie when he finally became an adult and sang. Okay, so you can hear, relative to those songs you just heard, that's a very different song. And you can actually kind of read these as you, much like a musical score. So I'm going to try this one more time. A bunch of short, and then those last two, that long note. So this is what we got. This is actually a sound spectrogram. This is frequency as a function of time. So what Leslie was doing there, those 2 last long notes actually are very much like his mother's contact calls. So when zebrafinches see other birds flying around, both males and females will go, mee, mee, and kind of try to lure the other birds to come and hang out with them. So his mom would do that when birds would fly by the window. So he actually incorporated this song of his mom's into his song. I shouldn't say song of his mom's, this vocalization of his mom's. Okay, underlying this behavior is a pathway, and we know the anatomy of this pathway fairly well. This is a schematic of a semi-sagittal section. So you all remember, you've all had neuroanatomy at this point, I presume, right? So you remember the sagittal would be going right down the midline like this. And this side is anterior, this side is posterior, so the beak is over here to the right. And the nuclei, and by nuclei I mean collections of cell bodies, are represented by these circles, and the axonal connections between them are represented by these arrows. And we know a few things. For example, it looks like this one up here in the telencephalon, HVC, is responsible for setting up the timing of the various notes in the song, and RA seems to code indeed for the various syllables, and this is the nucleus of the 12th nerve, which goes out to what they have by way of a voice box, which is a syrinx. We know if you make lesions in this part of the pathway, HVC, RA, or this 12th nerve, you'll disrupt the output of the song. So once they've learned it, if you blast out one of these nuclei bilaterally, the males will get all set to parade around and sing, and they open their beak and nothing comes out. If in contrast, in this anterior region, MAN, area X, or DLM, which is down in the thalamus, if you lesion these nuclei, the acquisition of song is disrupted. But once song is been acquired, the lesioning of these 3 nuclei doesn't have a huge impact. So, as I mentioned, there are differences in singing behavior. Females sing, males don't. And these sex differences in behavior are related to anatomical differences in the song system. And so what I'm going to show you next are a series of slides where we have cut through and the coronal plane. So remember, the coronal plane goes like this. So I'm going to show you, the first slide I'm going to show you is this purple line where we've cut through the coronal plane like this. And the second section I'll show you the coronal plane going through HVC, and then the third through RA. And I'm going to show you what it looks like in a male and also a female. So here we see the coronal section of going through the anterior part of the brain. And what you can see over here, this line here, if you're watching, this line here defines what is essentially the striatum. And you will recall that the striatum is made up of what? Caudate and putamen. All right, so caudate and putamen. And so there's a specialized set of neurons in the male that stains really darkly, this kind of teardrop shape of neurons that's called area X in their striatum. And this area is completely absent or quite vestigial in the females. They don't have it at all. All right? Now, the second slide I'm going to show you here, the male here on your left, this is through HVC. This is in the dorsal part of the telencephalon. This line right here actually is the lateral ventricle that's now collapsed because the CSF is all gone. But you can see that the male has this great big nucleus here, and in contrast, the female has this little tiny slip of a nucleus, HVC. And then lastly, now we're in the caudal part of the telencephalon, and this is way back, this for reference here on the midline is the cerebellum, so we're way back in the caudal part of the telencephalon, and you can see the male has this nucleus that's shaped like this, and the female has this very densely packed nucleus that's shaped a bit like a comet or a comma, if you will. So I've shown you here actually pretty dramatic sex differences in the brain. You can see some of these, like you can see area X without even a microscope, okay? You just hold it up to the light and it's obvious, okay, if it's a male or a female. And these slides are about, I would say 4x. This is not a huge magnification. These are really very big sex differences in the brain. And you can imagine if you could see them that easily, you could sure quantify them quite readily. So this is a slide from one of the papers in your reader. This is from Nottebohm and Arnold, and you can see area X, male, shown by this symbol here, and then females, again, don't even have one. All right, so obviously a huge sex difference. HVC, if you compared the volume of males versus females, you would see that there's about a 5-to-1 ratio. And similarly for RA, if you compared the volume of RA, males versus females, there's about a 5-to-1 sex difference there too. Okay, so things that are different in the neural song system is, of course, the volumes of the song regions that I've just shown you, the number of neurons in those song regions. If you would count them out and figure, you would see that males have more than females, the size of the neuronal somata. So by somata, I mean the cell bodies are bigger in males and females, and also the accumulation of androgens. So males will have cells in these regions that will be more likely to express androgen receptors than females do. All right, so to summarize what I've done so far in this lecture, I pointed out that zebra finches have a behavior that's sexually dimorphic. It's song. Males sing, females don't. And there's a neural circuit that mediates this behavior that's also markedly different between the sexes, between males and females. So the question that I've been addressing now for so many years is to figure out, as this male is asking, what makes their brains so different? And so what she is going to suggest by way of an answer is the organizational effects of steroid hormones. Now, before I jump into that, I'm going to first talk about what determines sex at a biological level. And the story I'm going to tell you is the mammalian story, because the bird story hasn't quite been worked out completely yet. So in mammals, there is a gene called the SRY gene. Have you all heard this story before? I'll take that as a yes and a no. Okay, there's an SR, it's all caps if you're a human, and if you're a mouse, it's SRY like that, okay? And this stands for the sex-determining region on the Y chromosome, all right? And this gene codes for a peptide that's also known as testis-determining factor, and by looking at this, it looks like it has aspects of this protein that would bind to DNA and cause changes in the regulation of DNA, okay, or regulation of other genes, and that's probably what it does. This SRY gene is found, surprise, surprise, in XY males and XX males. So some of you probably just hit a little bump and said, how can you have an XX male? All right, so in this biz, we determine what is male and female by what the gonad is, okay? So if the gonad is a testis, we call that person or animal a male. If the gonad is an ovary, we call that person or animal a female, okay? So yes, there are XX males. How the heck does this happen? One of the ways it could happen is you get a translocation of that gene off the Y chromosome onto the X chromosome. So you could have a double X that's carrying that SRY gene. And if she's got that SRY, he has got that SRY gene, it's going to develop a testis. Okay? Okay, this SRY gene is absent in double X females and some XY females. Okay, here's another speed bump. How can you have XY females? Okay, if that gene is deleted, then the individual will grow an ovary. Okay, and there's some XY females that have had a frame shift or point mutation, so that gene can't make a protein that's recognizable, or sometimes can't even be made, and so they end up developing an ovary and end up being female. Okay, so once again, so it's the SRY gene is there on the Y chromosome, and I don't want to leave you with the impression that it's all going on in the male and nothing's going on in the female, but this is what looks like it happens. This is in mice. So in blue, we have the expression levels of the SRY gene. That kicks off first. What's got to happen subsequently is the SOX9 gene has to go up in males. Okay? And if it doesn't go up, or there's something wrong with the SOX9 gene, you won't get a male phenotype. Okay, it won't develop into a testis. So there has to be some downstream things too. You got to have SRY, but the way I remember it is you got to have your SOX on to get a testis. All right, so SOX9 is also going to be on. If you don't have this SRY expression kick off, what will happen is in females there's another gene that will go off called DAX1. All right, and in males this gene will in DAX1 will be down-regulated in males, okay? So it's not that little girls are just doing nothing, but the one thing I want you to remember is that in mammals at least, SRY is the genetic switch, okay, for mammalian sex determination. The presence of the SRY gene is necessary, but not sufficient, okay? So you have to have some downstream things to go on. And that's for sex determination, okay? So I'm going to make a big deal here between sex determination and sexual differentiation. So sex determination is what's the switch to determine what the gonad will be, a testis or an ovary. Differentiation is the rest of the development towards a male or female phenotype. Okay, so as we've mentioned, first of all, there's genetic sex determination, which is kicked off by the presence or absence of that SRY gene. If the SRY gene is present, the individual is going to develop testes. If the SRY gene is absent, the individual will develop ovaries. After that, what looks like happens is, at least in the classic story, the rest of development is driven by the hormones that come out of the gonad or not. Okay, and by that, in early development, what we call the organizational period, the little testes in fetal life or in perinatal life, so peri means around, right? So either right before birth or just after birth in rodents. In us, all this happens in utero. The little testes are going to secrete androgens, okay? If that happens, you'll have developed primary sex characteristics. So the little baby will grow a penis, a scrotum, etc., okay? In contrast, the ovaries basically don't do anything in early life. Their secretions are very, very low. And so I wrote nothing here. And if that happens, then the development will be in the path of female characteristics. She'll grow a vagina, labia, etc., okay? And so at birth, the doctor will be able to look between the legs and declare this is a little boy or this is a little girl, okay? So that's the organizational effects of steroid hormones on the periphery. Again, driven by the gonadal steroids. Activational effects of gonadal steroids happen at puberty and on into adulthood. So activational effects are what happens in puberty, in our species and all others, those aspects of development that a lot of you are interested in scoping out in the opposite sex, okay? So women grow breasts, everybody gets hairy, men grow beards, everybody gets more muscles for reasons I'll explain later. So those are the secondary sex characteristics, and those are driven by the activational effects of steroid hormones, okay? So and I'll explain in further detail in this next slide a little bit more about what is organizational versus activational effects of steroid hormones. Okay, and in particular gonadal steroids, which is going to be the focus of this unit. So in organizational effects of steroid hormones occur early in life, so very early in development. What we're going to be looking at in the lab are some brains from birds that were treated immediately after they hatched, okay? And organizational effects having happened during a critical period. I'm still fond of this concept, although it has been criticized. Some like to say a sensitive period. But if I had given those hormonal treatments to the little babies 20 days after they hatched, wouldn't do anything to them. Wouldn't do anything to their development. If I do it immediately after they hatch, it's going to have a profound effect. And the organizational effects are relatively permanent. So these brains that we're going to be dealing with were taken in adulthood, and in adulthood we can see manifestations of the treatments that happened even when they were babies. So those the brains changed, and we're going to be able to see in adulthood the effects of the treatment of babies because it's babyhood because it's relatively permanent. Activational effects, again, are that which happens at puberty, goes on into adulthood. It's later in life, obviously, during puberty usually. There's no critical period, okay? So the activational effects can happen any time, okay? And the activational effects are relatively transient. So with birds, especially seasonal breeders, which are the birds wild birds we have around here, what you'll notice is, especially in these last few days, the birds have started singing again. Does anybody notice that? They haven't been singing for months, okay? Why is that happening? Well, because the photoperiod's about right, was warm there for at least for a little bit, and they're all going to be paired up by about the time St. Valentine's Day rolls around, which is one of the reasons why St. Valentine's Day is associated with love, is the birds pair up. So the males are starting to sing. Why are they starting to sing? Because their gonads are starting to grow. During the winter, since birds have to fly, if you don't have to carry around a heavy gonad, they shrink down. Okay, so the males' testes are starting to go up again. They're getting big androgen levels. Now they're starting to sing again and court. Okay, if we were to go and grab one of those birds, take his testes out, he'd stop singing again. If we gave him an implant, put his testosterone back in, he'd start singing again. Take the implant out, he'd shut up. Okay, so the effects are relatively transient. They're going to come and go with the hormones. There's no critical period. We could put them on the shelf for a year or 2 years, plop a pellet in there of testosterone. Wouldn't matter when we did it. He'd start singing. Okay? No critical period activation. Our study is going to focus on organizational effects. Okay? I just want to contrast it with activational effects. Okay, let's talk now about what are the steroid hormones, and I must admit I don't usually like Wikipedia. but I got this diagram from it, and I don't usually like charts with lots and lots of stuff, but this one actually is a gem, and I was really happy to run across this. So these are the steroid hormones, and the mother of all steroids is cholesterol, and one of the things I really like about this particular chart is they show you the scheme by which they numbered all the darn carbons, okay? And that's how these things get named by and large. So that actually helps me out. So cholesterol gets converted, first of all, into progestins. Progestins are shown here in this sort of olive drab box. And I'm not going to deal, these are adrenal hormones out here. I'm not going to talk much about those. I'm going to talk about the gonadal steroids. But you can see the progestins actually get transformed into the gonadal steroids. And here in this gray sort of L-shaped box, we have the androgens, okay? And here's one. Anybody ever hear of DHEA? No? Well, you can go and buy this stuff now by the bucket in health food stores. It's supposed to be great for you. It's not regulated. I don't think it probably has very much effect unless you're to take really high doses, which I wouldn't recommend. DHEA gets converted. All the enzymes, by the way, are in these green boxes and named. And so it gets converted into one of the androgens that has a bit of a biological punch, and that's androstenedione, okay, androstenedione, okay? And that gets converted into testosterone, and testosterone has a big biological punch. Why is that? It has a big affinity for the androgen receptor, and it can really kick off androgen-related events, okay? And similarly, it can also be reduced into this one, dihydrotestosterone, DHT, and that has a big biological punch too, okay? Mostly in the periphery, it's DHT. Oh, by the way, neuroscientists have this really awful chauvinistic way of calling the central nervous system is special and everything else is periphery, right? So out in the rest of the body, DHT is probably primarily responsible for masculinization, okay? Now, we're used to thinking of androgens as being male hormones and estrogens as being female hormones. But I can tell you, if we were to take measurements right here, every man would have measurable levels of estrogens and every woman would have measurable levels of androgens, okay? No sex has got an absolute lock on androgens or estrogens. But there are differences in the levels and differences in the pathways that are favored by women versus men. So everybody is going to get this androstenedione, okay? And this, as I said, is a relatively weak androgen, but it's going to be present both in men and women. And this is what gives us things like armpit hair and hairy legs and stuff like that. But this can be converted to an estrogen. So in this triangle here, this gray triangle, we've got the estrogens, okay? It can be converted first to estrone and then to estradiol. Now, estradiol is the estrogen that has the big biological punch. It's the one that has the strongest affinity for the estrogen receptors. Estrone's affinity is somewhat weaker, and so is estriol's, but they're all estrogens. But estradiol is the one that can really make big biological changes in the body. And again, it's due to its affinity for the receptor, which is a protein. I'll be talking about that in a minute. You see in the popular press about good estrogens and bad estrogens and things like that. I actually think that's a bit of hooey because they say, well, the estrone is the good estrogen and estradiol is the bad estrogen. Well, it's just a matter of the affinity for the receptor. So you could probably get the same effects out of estradiol depending on the dose. All right. Before I go to my next slide, all of you had organic chem, right? All right. Are these molecules polar or nonpolar? Nonpolar. All right. So these are greasy molecules, right? Okay. Do you think you could dissolve these in water? No way. You'd be there from now to almost eternity. All right. Since they're nonpolar, the way they act is like this. So this little thing's going to represent one of my steroids. And since it's nonpolar, it does not need a receptor site on the cell membrane. Okay, the cell membrane is primarily made out of what? Phospholipids. Okay, and since it's all that lipid, it's just going to slide right through there. Once it slides right through there, what it will usually encounter actually out here in the cytosol instead of on the membrane, like other receptors you might have heard about, is a receptor that is specific to that particular hormone. So if it's estradiol, you'll have an estrogen receptor, and those will bind, and then this hormone receptor complex will bind to the DNA and affect some changes there in the DNA, okay? Both in development and in adulthood. Okay, so this is one of the other big points of this lecture, and this is the story that you're going to find in every textbook, and that is that sexual differentiation of the brain is driven exclusively by gonadal steroids, okay? And how did we get to this story? It was from this magic moment. All right, so this is a sexually dimorphic behavior, and almost all this story came out of studies with rodents. Okay, so obviously this is copulatory behavior. So I'm now going to ask the cameraman, I'm going to have you put the camera on me instead of the slides. Maybe I'll stand over here so I'm more easy to see. So this behavior is very sexually dimorphic. Males engage in the copulatory behavior, and if you saw it, you wouldn't think it looked anything like sex. But what the males do is they will mount the female, or they will intromit, and if they do intromit, what they do is they hop off like that, and lastly, if they intromit and ejaculate, then they'll palpate her flanks like this. And mice are very cute. They'll palpate her flanks and then really hug her, and then they all fall over after he ejaculates. Okay? So females never do that stuff. Okay? So it's a very sexually dimorphic behavior. Females, for their part, if they're not in heat at all, they will just keep their rumps down, and if the male tries to mount them, they'll turn around and box him off, okay? If they're slightly in heat, they'll stand up. If they're really in heat, like this one here, what they'll do is they'll show this lordosis behavior, this big arch back, and they'll throw their head back when he mounts, okay? So you can score this behavior, and again, the males never do this, okay? So it's a real sexually dimorphic behavior. Males do one set of behaviors, females do the other. They almost never, ever do the behavior of the other sex. Okay, put the camera back on the slides. All right, so this is a slide that actually summarizes piles and piles and piles of research, okay? So what was discovered is that if you take a little female rodent pup, she's got an XX genotype, you leave her alone, when she grows up, she's going to get her sex hormones, her estrogen and progesterone, right? And so she will show that lordosis, that arch back and stuff like that, okay? If you take this same little girl, this little female rodent pup, and you give her a big blast of testosterone around just after she's born, even though she's got an XX genotype, when in adulthood you give her estrogen and progesterone, she won't do that typical female behavior anymore. And instead, if you give her androgens, you give her testosterone when she's an adult, she'll mount and act like she's intromitting and act like she's ejaculating. Okay? So her behavior will be completely flipped. Okay? Same thing for little males. You have a little male here, his testes secrete androgen when he's little, XY, you leave him alone. After he goes through puberty, he's going to get his testosterone, he'll act like a male. He will mount, intromit, and ejaculate. If on the day of birth you castrate this guy, even though he's got an XY genotype, in adulthood you give him testosterone, he does not act like a male. He doesn't show that mounting behavior. If you give him estrogen and progesterone, then he will act like a female. All right, and show that in response to a male mounting him, he will act like a female. All right, so you can, again, completely flip the behavior with the hormone treatment in early life. Okay, with manipulating the hormones in early life. Okay, so that led us to think that the effects of hormones are epigenetic. Okay, epigenetic, so that means above the genome. So happening apart from the genome. So that made us think The genome determines the gonad, and after that, the differentiation of the brain is driven exclusively by gonadal steroids, okay? And that was based first on behavior, okay? So we think it's completely gonadal steroids that determine the sexual differentiation of the brain. And what also led us to that conclusion is some studies that were done here, good old UCLA, right down the hill in the medical school. And so what I'm going to show you here, this is the pre-optic area. And as you have all told me, you guys are now whizzes at neuroanatomy, having survived Dr. Scheibel. Yes? Okay, so here is the anterior commissure. Here is the third ventricle. Here's the optic chiasm. So we are just anterior to the hypothalamus here, all right? And if you see the males have this big, dark patch of neurons here. And females, in contrast, have this little pinpoint of dark staining neurons. But if you give this little female early in life, like in the first 5 days, if you give her a blast of testosterone or estradiol, then she will develop this preoptic area looking like a man, or looking like a male. Not a man, a rodent. Okay. Why did estradiol work? Actually, it goes the other way. So let's go back to my handy little slide here. And what you can see here is that testosterone can get converted to estradiol. Okay? It's a one-way reaction, too. Testosterone to estradiol. All right? And so, to our great surprise, when these experiments were first done, estradiol worked to drive the brain in a masculine direction. Something I'll be coming back to. Okay, so summarize part 2 of this lecture. Again, one of the big points is sexual differentiation of the brain is driven exclusively by gonadal steroids. And I'm going to tell you right now, that's the story that's in all the textbooks. By the time I get done with this story is going to need a few addenda. All right? But that's what people have thought, and many people still think. Okay, so now what we're going to do is that we're going to take that central dogma and go back and look at zebra finches. And so what we're going to do is look at a study that is in your reader by yours truly. And so we're going to ask the question, which steroid hormone masculinizes the song system in these zebra finches? And she's going to say that her brain will be the model for his. So one of the strategies in this field is to give hormones to the female that ostensibly she wouldn't have in nature and then look at the consequences. So what hormones did we decide in this study? We decided to give equal doses of estradiol because of what went on in the mammals, testosterone because there were these guys at Caltech over the hill that said testosterone would completely sex reverse the song system. We wanted to check out and see if they were right. We gave androstenedione because this hormone had been shown to have the biggest effect in activating the behavior. So we thought, well, maybe an organization that's special. And then a friend of mine who'd mapped out progesterone receptors convinced me that I wanted to look at progesterone too. So I gave equal doses to all the zebra finches, and we gave it to them as babies, okay, because again, we're looking at the organizational effects of these steroids, right? So we gave them an implant. Anybody ever hear about Norplants? No? Okay. Well, it's a kind of implant that's used with humans. Basically, we made our own zebra finch size. This is a little zebra finch baby. This is its head with a little bit of downy feathers there. That's a foot, and there's a pencil for scale on these things. All right, so we're going to give a little tiny implant to these babies, and what we found out was this. So here is one of our dependent measures was the volume of that nucleus RA, and here we've got males and all these different other groups or females that were treated in various ways. The blank is the control, so they got a pellet with no hormone in it. All right. So in this study, you can see we got our sex difference, replicated our sex difference. That's always important. Males were larger than females. We saw that estradiol actually masculinized this song system quite well relative to controls, and so did testosterone. But estradiol actually worked better than testosterone, okay? Even though testosterone worked. So why would you think that would come out like that? Why would it come out like that? Equal doses of all this stuff. Okay, testosterone can get converted to estradiol, but remember, it can also get converted to DHT, or dihydrotestosterone. So it gets converted to estradiol, but not with 100% efficiency. So you don't get all the testosterone ending up being estradiol. All right. So probably the reason it worked at all is it got converted to estrogen. All right, some of it. Okay. So this guy says, so does estrogen masculinize your song system more than testosterone? Is this a weird bird thing? And no, it's not. In fact, it's been seen in mammals too. If you do a side-by-side comparison, same doses like I did here, estrogen works better than testosterone to masculinize in mammals too. Again, because in neurons, testosterone gets converted to estrogen. Okay? Okay, so now he's going to ask another study, and that is, he's going to ask another question, which is your brain exquisitely sensitive to estradiol in development? And this is actually the question that you are going to be addressing in the lab. And so she's going to say, okay, let's go ahead and see. All right. So what we're going to be doing in lab is we are going to be doing a dose-response study of masculinizing effects of estradiol. Estradiol, by the way, gets shorthand is E2. Okay, so sometimes you'll see me write that up for estradiol. Okay, so one of the very first things is you guys are going to have males and female controls. We want to see if we can replicate the sex differences. If we can't replicate the sex differences, then is your study going to be very valid or worthwhile? No. You can't replicate the sex differences and show you can at least get the phenomena, then you can't start talking about what could the phenomena be due to. All right. The next thing we're going to do is we're going to gauge the sensitivity of the female song system to estradiol, and we're going to do that because you'll have females getting different doses of estradiol. Okay? What we're also going to see is masculinization an all or none event? That is, once you get to some threshold value of estradiol, does it all just kick off and masculinize, or does the masculinization go up with the dose of estradiol? Okay? Okay, we're also going to find out are the different parts of the song system equally sensitive to estradiol or not? Okay, or does one masculinize and another doesn't? Something like that. Okay, we'll be able to tell that, and we're going to be able to see if any of our doses fully sex reverse the females. Okay, into being as masculine as the males. Okay, make sense? Yep. All right, so our groups explicitly, we're going to have, again, a group of control males. They get implants, but there's no hormone in their implants. And I have to admit, I already did this for you long, long ago because it takes them at least 90, 100 days to grow up, and we get 2 weeks. So this has already been done. Okay. You're going to get a group of control females. They also got implants, no hormones in their implants. Okay. One group of females is going to get an implant with 5 micrograms of estradiol. Another group is going to get 15 micrograms of estradiol, and the third group will get 50 micrograms of estradiol. And one of the reasons why I did that was in the literature, everybody said, oh, you have to have at least 50 micrograms to masculinize this system at all. Otherwise, it don't work. But as I started looking around, that was something that everybody believed, but nobody had ever tested. So we were going to test that and see if that's true or not. You all are going to be blind when you're making your measurements on these various brains. What does that mean? You don't know who's in which group, right? You're going to know who's male or female, who's got 5 micrograms, who's got 50 micrograms. You don't know. Why am I going to do that to you? Seems awful. Yeah, because I know you all want to please me, desperately want to please me. So if you knew that that's a male and this other one's a female, of course you're going to draw it bigger just to make Dr. Grisham happy, aren't you? Of course you would. So if you don't know, you can't do that. All right? And that's actually a really, really important control. You got to make sure that you're blind, okay? And you're going to be blind. All right? So you're not going to know who's male, who's female, what dose they get. All right, so what you're going to do is you're going to replicate and extend an experiment that I already did, and you actually have the paper. I was lucky enough to get it published a couple of years ago. And it's based on the dogma that gonadal steroids are responsible for the sexual differentiation of the brain. And we're going to look at the organizational effects of estradiol. So all of these birds were given these doses when they were babies, little tiny hatchlings, and we let them grow up to, I believe, 100 days of age before we sacked them. But the manipulation was when they're babies, so it's organizational effects, okay? And you're also going to extend my study, because in my study I did not have any males. I just had all females. So you're going to extend it a little bit, put some males in, and see if we can sex reverse those females. All right, so you're going to check that. All right, so to summarize what we're going to be doing in this module, Lecture 1 was some background to all this, what we're going to do in lab. We're going to measure the bird song system, and you guys, all the brains have already been cut and photographed, but I'm going to give you the chance to section one of these brains yourself, a mystery brain, and I may not even know what sex this mystery brain is, but once I look at the slides I'll be able to tell. So you're going to section a mystery brain, put it on slides, hopefully not shredding the tissue as you do that. It's not like falling off a log, let me tell you, but at least you get a chance to try that. Lecture 2, I'm going to give you some more background. Why was this study interesting to do in the first place? Lecture 3, I'm going to tell you how to write a lab report, part 1. I'm going to ask for a little more formal lab report than Bolter and Weinmaster, so I'm going to ask you to have like an abstract, intro, methods, results, discussion, just like a journal article, more or less, okay? Lab 2, what we're going to do, finish measuring our brains. We're going to analyze the data, which I hope will work out beautifully for you all. We're going to stain and examine the mystery brain, figure out what sex it was, hopefully. And then lecture 4, we'll talk about your data and how to finish your write-up once we get your data. All right? And I'm just about right on time, so on behalf of the birds, enjoy the remains of the day.